So we are live now. Good evening, participants. Welcome to the live session of the course Basic Genetics. Our course instructors, Professor Richa, Arya ma'am, and Professor Jagat Roy sir are here with us. Participants, kindly feel free to join in and clear all your doubts. First, we are going to address the queries raised via Google form. A student named Hemant Kumar has posted a query. The query is, what is the key difference between base substitution and frame shift mutations in terms of their effect on proteins?
Yes. Is Hemant around?
No, sir. Right now, only one student has joined, Ms. Tanisha Gupta. The rest of them are yet to join, sir. But since this session is getting recorded, the students can watch the event later on also, sir.
Okay.
Yes.
So when Hemant joins, then only this will be answered, isn't it?
So you
Not
can
like
answer
that, sir.
right
You can
now.
answer it, sir. You can answer it, sir.
Okay. So difference between base substitution and frame shift mutation. Base substitution means one base is substituted for another. So here, the thing is that nothing will happen to frame because the frame remains the same. And when you, so what happened that here one base, let us say, is removed, another base is added. So that is what is substitution. And frame shift means one base is added where nothing is being removed. So what happened if it is a three base codon? So these three base codon is now shifted because one base is added. Let us say you have 6 bases. So that means you have two codon, codon 1 and codon 2. Now one base is added. Result is codon 1 and let us say in between another base is added. So now the total becomes 7. So 3, 3, 1 extra. So that means here the amino acid will get changed because in between one base is added. So here the amino acid will change from that point where the base is added. All other bases, all other amino acids will get changed because one base is added. Now, let us say it is ATG, then TGA. Let us say this is the sequence. When one base is added, let us say C is added. Now, in place of TGA, now it has become CTG. So that means this is coding for another amino acid. And subsequently from there, all other amino acids will change. But if it is a base substitution, then that base is, like one base is added in place of another. One is removed and one is added. So result is that particular amino acid will change. Rest everything will remain the same.
Thank you, sir, for addressing that question. Shall we go on to the next question, sir? So the next question. So can we move on to the next question?
Yes, yes, please go.
Yeah. So the question is, could you explain how epistatis differs from dominance? Sometimes the definitions seem quite similar. This was posted by a student, Tejas Goyal.
So actually, I think sir is a bit busy. I can address that. So see,
Sure.
epistasis, when we are using word epistasis, this is when we are talking about interaction between genes, right? So when two or more than two genes are interacting with each other, and we want to understand the relationship between these genes, that is called the epistasis. Whereas when we are talking about dominance, dominance is the allele. So you have capital T and small t. You have tall plant and dwarf plant, right? And those are the same gene, but two different alleles. And out of the two allele, which is showing the effect is called dominant allele. So in case of capital T, small t, or tall or dwarf, tall is dominant over dwarf. So these two are two different things. One is for the gene interaction or effect of the two alleles, and then in epistasis we are talking about more than two genes. Interaction between or amongst the gene is epistasis. Sir, please add if you think there is more to be added to this question too by Tejas. Sir,
Jagat sir, would you like to add
would
anything?
you like to add anything? Madhu ma'am
It's
has
fine.
also joined
Yeah.
actually.
Welcome, Madhu ma'am. Your mic is muted, ma'am. We received few questions from our students. We are addressing those questions, ma'am. Your mic is muted, ma'am. Richa ma'am, can we move on to the next question?
Yeah. So do you want to add something to the question too? I actually I addressed. We can move to the next question. Yes.
Yeah, sure, ma'am. So the next question is posted by, it's a set of questions posted by Ms. Savita Meena. Let us look at the first question. The question is, why do we need multiple types of banding techniques if they all show chromosome structure?
Ah,
Yes, Roy, sir.
answer to this is,
Yes, sir.
means we are talking about different types of banding. The first banding that was done, that was G banding. G banding gives you the light region and dark region. So you have a chromosome subdivided into different regions. And this region actually reflects the inner architecture of the chromosome. That is how tightly, in a chromosome, the DNA is tightly packed, at some places slightly loosely packed. Again, tightly packed. So tightly packed regions gives you G-band positive region, and loosely packed regions give you G-band negative region. So this is G-banding. This is the first banding, and that gave uh, that can be used in the nomenclature very well. The other type of banding, there can be another kind of banding, which is reverse banding. So in reverse banding, this is exactly opposite of banding, G banding. Why it is used? Because if suppose somebody is having a fluorescence microscope, somebody is very happy with the fluorescence microscope, and this is simple stained, chromosome stained and seen under fluorescence microscope. So that is why some people do the reverse banding, or not reverse banding, the Q banding. This is quinacrine mustard banding. So G banding, means somebody may ask, why not G banding only? In G banding, when you do the banding, what happens? You treat with trypsin. Sometimes chromosome lose its architecture if the trypsin treatment is a bit more. So this is, you have to control in a better way. But if that is controlled in a better way, G banding is the banding on which the entire nomenclature is dependent.
Thank you, sir.
So
Can we move on to
once
the next?
again,
Yeah.
the G banding is the main type of banding. Q banding, one can do if one has a fluorescence microscope and one is very easy with the fluorescence microscope, then one can go for that. The other banding is C banding. C banding purpose is totally different. The purpose is not having the different regions. It is not having the different regions of the chromosome. It is the centromeric region or the telomeric region of the chromosome. To see the centromeric region, to see the telomeric region, that is what is C banding. This is called the constitutive heterochromatin banding. And why this is done? You may say that why should we look for only the heterochromatic? Why should we only look for the heterochromatic region? Only heterochromatic region can, by that also, you can see the different structure of the heterochromatic region or the different structure of the chromosome centromeric region. And on that basis also, you can identify the chromosomes. But it is not that chromosome can be banded or chromosome regions can be made clear. So this is the answer.
Thank you so much, sir. Can we move on to the second question, sir?
Yes.
Yeah. The question is, what is the logic behind the nomenclature of bands in a human karyotype?
The nomenclature, it is like this, that now a chromosome has been subdivided into different regions because you have the chromosome in the form of G banded. Just I take 1 second. Hello?
Sir, your mic is muted, sir. Sir, kindly turn on your mic, sir. Mic is muted, sir. Mic is muted, sir.
Yes.
Yes, sir. You're audible now,
Now,
sir. Please go
what
ahead,
is
sir.
the logic behind the nomenclature of bands in a human karyotype? One wanted that if suppose you stain the chromosome and if you observe, many chromosomes look alike. Many chromosomes size-wise, shape-wise, particularly the group C chromosomes. You have a large number of chromosomes, that is chromosome number 6 to chromosome number 12. All are almost having similar size, similar shape. That means on the basis of the shape, that is metacentric, submetacentric, acrocentric, or on the basis of size, you cannot really identify which is which. That means it is very important for you to identify chromosome. Unless you identify chromosome, nothing can be studied further about that chromosome, or let us say if if let us say some kind of anomaly is there, one cannot identify. So that is why chromosome identification is very important. And for that, if suppose G band is done, suppose if G banding is done, then each chromosome is having subdivisions. And now these subdivisions, if they are numbered, then you know the position of different subdivisions of a chromosome. For example, if suppose you have chromosome 1 has 2 arms, P arm and Q arm. If suppose now I am referring to band number 1 of P arm, that is the top side or the shorter side of the arm. So there the band number 1. So how you will write? You will write 1, chromosome number 1, P, and band 1. So this way that particular region is focused, that this is the region of that chromosome. If suppose now on the P, we are going for band number 2, then we will say 1P band number 2. So that means another region of the chromosome is now being identified or being studied. There are, means this can be further subdivided. Further subdivided means the mnemonic, when you have banded the main bands, and there are finer bands within the main bands. So that means band number 1 is having subband 1, subband 2, subband 3. So it becomes 1, 1, 1, 2, 1, 3. So main thing is the entire chromosome you have subdivided into different regions, and thus any anomaly or any kind of difference is there in a chromosome that can be identified clearly.
Thank you, sir. Can we
Okay.
move on to the next question, sir?
Yes, just give me one more moment.
Sure, sir. Sure, sir. So Madhu ma'am has joined. Good evening, ma'am. Welcome to this session. Your mic is muted, ma'am.
Is there a question for me?
Yes, ma'am. Richa ma'am has identified one question, the last question for you. Once Jagat sir
Should I-
answers.
Okay.
Yeah. So just a second, ma'am. Sir, I'm just reading out the third question.
Yes, third one.
yeah.
Can I answer the third one now?
Yes, sir. You can answer the third
How
question.
does
Sir.
infection by HPV actually lead to cancer development? So here, the thing is that human papilloma virus, this is a DNA virus. Let us say this has infected an epithelial cell. This is epitheliotropic virus. So it infects the epithelial cells. So infection occurred, and then this virus, the genome of the virus that has the different genes in it. So some of the genes that is responsible, those you call it as oncogenes, and they are responsible for cancer development. So that is how the cancer develops. There can be two things. One is HPV virus infection, and some of the genes of the HPV virus, this is responsible for the oncogenicity, and thus cancer develops. So this is the answer.
Thank you, sir. Richa ma'am, can we move on to the next question posted by Asif Nawaz? Your mic is muted, ma'am.
Yeah. So the
Yeah.
student is actually asking about the concept of transduction. And yes, as he wrote, that it is indeed fascinating because it's the infection of a bacteria. Bacteria is getting infected by virus. And this mechanism, as he is asking, that, it is also used for the mapping of bacterial genes. So if you definitely know, we have discussed in our classes that when a virus infects bacteria, right, then what it normally does, it injects its genome into the bacterial cell. And then there are two options. One is that it just completely lies the bacterial DNA, and then take the control and use the whole cell, emptied cell to amplify its own DNA and then come out, right? And so in that process, it can also pack the DNA of bacteria into its capsid. So that is something which is useful, because if this virus packs the bacterial genes into its genome and then it infects the next bacteria, then it transfers those genes into the that bacterial cell, and that can be used for the mapping. And as it and this the student is also asking how accurate is the bacterial gene mapping compared to the eukaryotic mapping. See, this question has two angles. Now, if he is asking in terms of transduction or the standard mapping methods, that where we are using the gene mapping methods by recombinations, or in case of bacteria by gene transfer, and then how they are transferring to the next bacterial cell, compared to those that are recently available, which is sequencing, right? So sequencing is both the same, right? For both, you can just sequence the genome, so it's same for both. And as far as the accuracy of the bacterial gene mapping is concerned, they're pretty accurate. The only thing is that you need to have different overlapping methods to confirm, the minor differences that might be present. So, as far as I understand, these methods are accurate, and that is how these concise maps initially were generated. And now, well, when the DNA sequencing is available, we also have the genome map, the genome sequences, where we clearly know which base is where and which gene is where. And
Thank you, ma'am. Can we move on to the next question, ma'am?
next question, yeah.
Yeah, so the next one is posted by Miss Ishani.
Yeah.
The question is, how does genetic counseling help families understand the risk associated with hereditary disorders?
Yeah. So actually, when we do the genetic counseling, what exactly happens is that patient, or the person, actually, rather I should say, a person present with some problem, and it's the job of genetic counselor to identify if that disorder which somebody is presented with is having a genetic basis or not. And to do that, you need to collect the family data. You need to know their siblings, and their parents, then their grandparents, and the extended families, how that particular trait is running in the family. And if you identify that, then you can certainly sensitize the family to know, what are the chances if they are planning a baby for having that disease in their case, right? And secondly, if there are options available which they can take, before birth, then they can do that. And if there are other, like in case of for example, I think I probably have given that example of thalassemia. Like, thalassemia is a disorder which is recessive, and then if genetic counselor identifies that the parents are having, and they are planning a baby, they are having recessive allele of thalassemia, then they can tell them that the baby could have a 25% chances of having the disease. And then they can get the baby tested by a mother. Definitely a pregnant mother can be tested for the presence of disorder in the baby, and then they can opt for the options available to them. So genetic counselor is somebody who really can assist people understand about the disorder, their consequences, and their options that they can use to treat these, or to not treat, tackle these diseases. Sir, do you want to add something?
Yes, I'm talking about genetic counseling. Just 1 minute. One of my colleague and teacher, Professor Rajiv Raman, he does a lot of genetic counseling. I give you one example, means how effective it is.
Hmm.
One day, somebody died, and we had, myself and Professor Raman had gone to his place for just saying sorry, and sorrow, expressing sorrow. There we were sitting, and one photo of a girl was there on the wall. So Professor Rajiv Raman asked him that whose photo is this?
Mhm.
That person said, this is my daughter. Said, why this photo is like this decorated? He said, she is no more. What had happened? He said, I do not know. Suddenly one day she died. And he said that today his wife also died very suddenly. Immediately, Professor Raman, he said, okay, let me see the hands and feet of the dead body, and let me see if you can provide a photograph of the girl whose picture is there to see the hands and feet. By seeing that, immediately he said, this is Marfan syndrome. And because of these kind of deaths has happened. Today you do all rituals, everything. Tomorrow, the entire family should be brought before me, and I'll counsel. And after today, there will be no death. I can guarantee you. So that is the power of genetic counseling. Okay.
Beautiful example.
Thank you, sir. Madhu ma'am, would you like to add something, ma'am?
I think Professor Roy and Dr. Richa, they have explained it very nicely. And the basic function of genetic counselor is, of course, to identify where the problem lies and convey to the parents in layman's terms so that they can understand.
Thank you, ma'am. So can we move on to the next question, ma'am?
Yeah.
Yeah. So the last question was posted by Mr. Chandan Mehta. The question is, I'm a bit confused about quantitative inheritance. How do we statistically distinguish it from simple Mendelian inheritance?
Okay. So by reading this question, it's difficult for me to actually understand what the student wants to ask. But what I have understood, I'll just tell. In case there are more queries, then the student can of course approach. So when we are talking about inheritance, so whether it is quantitative or discontinuous inheritance, all are Mendelian inheritance, even quantitative inheritance, the genes and the alleles, they follow the same Mendelian laws. They follow the law of segregation, they follow the law of individual assortment. So there is no difference in the quantitative inheritance following some other method. So they both follow quantitative. So quantitative inheritance is actually continuous inheritance, and the traits that generally we talk about in Mendelian inheritance, they are discontinuous inheritance. Both follow Mendelian laws, and statistical tests can be applied. Same statistical tests can be applied to both the simple Mendelian crosses, monohybrid, dihybrid, or quantitative traits. The only difference here is in quantitative traits, we have to identify the QTL, quantitative trait loci. That means all the loci that are present in an organism that are responsible for imparting or those are important for this particular quantitative trait. Once we have understood the QTLs, then one can pick up any statistical analysis. There are a lot of statistical softwares these days which can be used. And the second thing is in quantitative quantitative traits, for example, for traits of human disease like blood pressure or heart problem or diabetes, there are certain SNPs, single nucleotide polymorphisms, which are associated. So we have to do a kind of genome-wide association studies. That means you associate a particular SNP with the disease. And then so you have to have a large number of subjects, where you can study a particular SNP, whether it is related to a disease or not, and then you can do there are a lot of algorithms that are available, softwares that are available, where you can put your data and then you can understand. So it is not about statistically distinguishing between Mendelian or quantitative inheritance. Both follow Mendelian inheritance. In both of them, statistical testing can be done. The only difference is, depending upon the sample size and what questions you're asking, you have to apply different statistical tools.
Thank you, ma'am, for answering that particular question. So I'm just
Can
stopping.
I add
Yeah,
little bit to
sure.
this?
Sure, sir. Sure, sir. Please go ahead, sir.
Just I want to add that in simple Mendelian inheritance, what you study usually, this is one gene. You are talking about a monogenic trait. And in quantitative inheritance, it may be more than one gene involved. So if it is more than one gene involved, then it is not so simple that it will follow 1:2:3, or 1:2:1, or 1:3, that kind of a thing. But this will vary with the entire population has to be taken together, and the variation will be there. A lot of variation will be there because there are more than one gene involved. So that is why the quantitative inheritance little bit differs, but madam has very accurately said that both are actually Mendelian inheritance. Only difference is, means when we say a monogenic trait versus may not be a monogenic trait, but showing variation.
Thank you, sir. Richa ma'am, would you like to add anything to this particular?
Perfect. They are experts.
Absolutely, ma'am. So I'm just stopping the screen share. One student is here with us. Ms. Tanisha Gupta, can you unmute yourself?
Yes, sir.
Please go ahead. If possible, you can turn on your video also and interact with our instructors. So do you have any doubts?
No, actually, I don't have any query right now.
Okay, fine. And then examination for this course is on 24th of October. Ma'am, could you give some tips on how to prepare for the examination effectively? So it will be beneficial for the students who are appearing for the examination. Any one of you, ma'am?
No, I think definitely for any examination, revision is the most important thing that one should be doing. And I think this SOM platform has really nice way of providing, the video lectures, short video lectures that you can always watch. And then every lecture has questions associated with it. And then you have these transcripts. We have also linked the references. So read it, and then definitely if you have any doubt, you can write any of us. You are writing, you have these platform, swam portal platform, and I hope that we are constantly responding to student query. So the genetic course is definitely a course where one has to engage as much as possible, because more reasoning is there and more logic is there. So do your revision very well, and I'm sure that you all will do great in your exams.
Thank you, ma'am. Since there are no more queries, can we conclude the session, ma'am, sir?
Perfect. Yes, thank you, Murliji. Actually, before you thank us, I thank you. You're really very nicely organizing all these sessions timely and very meticulously.
Most
Thank you.
welcome, ma'am.
Thank you
Most
for
welcome, ma'am. Yeah. So thank you so much, Jagat sir, Richa ma'am.
I also thank you, Dr. Waldi.
You're
It was
most welcome.
really
Most
wonderful. And the way cheerfully you handled the things, that is very.
Yeah, so much work, actually.
Yeah. So once again, thank you so much, Jagat sir, Richa ma'am, and Madhu ma'am for this live doubt clearing session. You all have explained the answers in a very detailed manner with a lot of examples. It was very useful and informative. And We hope our students watch the YouTube recording and benefit from it. So we are concluding the session now. Thank you, everyone. Thank you.